Thank you, Antoinette. Our next speaker is Dr. Matthew Silva. Silva is the Chief Executive Officer at In Vitro and leads a strategic vision and mission to support the drug discovery and development community. Matt holds a PhD in biomedical engineering from Worcester Polytech Institute and brings more than 20 years of diverse imaging experience from pharmaceutical and contract research organizations. Prior to In Vitro, he was Senior Director of Translational Biology at Vertex, where he ran the imaging and histopathology groups, and previously he led imaging groups at Amgen, Millennium, and Takeda Pharmaceuticals, focusing on a broad use of imaging biomarkers to support drug discovery and development. Matt, up to you.
Good morning, everyone. Thank you, Steve. Thank you, Antoinette. That was a great introduction. Thanks for everyone getting up so early, and hope you had safe travels here. I'm here to talk about, so now we've been introduced to the instrument, and so I'd like to talk a little bit about the applications. I think it's really important for us as a community to recognize that great instrumentation's available to us, and we collectively will build those applications that validate it and that continue to advance it into the future. So this is a bit of a survey of some of the work that we've supported through the years. The general synopsis here, although the technology's been around for 5 to 7 years, is that like anything in medical research, it's still it's emerging. It takes time for it to develop and for people to really understand how to utilize it in daily research. And it is a great complement to existing tools that we have today, both pathology and radiology tools that we use in small animals. I'm going to focus on some applications really around biodistribution, protein imaging, and in gene, RNA, cell-based therapies, where there are some really critical questions that are left to be answered. And what I hope to accomplish today is not to introduce you to the technology, that's been done, but introduce you to some applications that will get us all thinking about how to further this technology. As we think about that future, it's the developments that we make that will create additional improvements in that technology. I think one example for us to think back on is how FDG and PET, that technology is almost synonymous to most clinicians. PET is FDG imaging. It's the vast majority of what they use PET for. But that technology has allowed advancements in chemistry and better understanding of biology to also develop some very important additional tools. So we see right now the amyloid tracers playing such an important role in the characterization of anti-amyloid therapies in Alzheimer's, and congrats to the folks at Eisai and Biogen for the recent data on lecanemab. Really excited to see what that imaging data looks like, but we know that it was a critical secondary endpoint in that study. And also congrats to the folks at Lantheus and Telix for their development of PSMA diagnostics that are going to be useful to better identify patients that are suitable for treatment with metastatic prostate cancer. So imaging certainly it's a great time for us as a community, and as these new tools come together, we're here to develop those applications. So the basic premise, why am I focusing on these two areas? Well, and I'll show you a figure in a moment that proves this, but biologics accounts for about half of the drug development pipeline nowadays. So it's very important that we help to characterize biodistribution and the effects of those therapies. And there's been an incredible amount of investment and new company formation around gene, cell, and RNA-based therapies. And one of the most critical questions, if you have an opportunity, John Maraganore, who was the founder of Alnylam and the outgoing CEO, did a great piece in Nature where he talked about the history of Alnylam. And he uses the word delivery 35 times in an article about the founding of a company, including 3 times in the sentence that from the beginning, it's all about delivery. So you cannot just inject these things into the bloodstream and hope for the best. So we're here to help identify where these things go and what do they do when they get there. And just to, this might be difficult to read in the room, but the folks at Pharma Projects every year put together a great publication on what's happening in the pharmaceutical industry. And so this is just to support the claims that I just made that about 45% of drugs in development are biologics, and that since 1995, certainly a hockey stick in cell and gene therapies. So what does in vitro do? And I think in vitro is maybe representative of the community here at large. We're here to help to develop new drugs. One thing that we all do that's not represented on this slide is we also characterize disease. And we characterize animal models and how well or not well they recapitulate human disease. But in addition, if you think about just the drugs we're trying to develop, what are the questions that we're asking? We're asking, where do they go? What is the biodistribution? We're asking about the pharmacokinetics. How long does it take to get there? How long does it stay when it gets there? We're asking a term that we use internally, maybe a gray area between pharmacodynamics and expression, but we talk a little bit about pharmacoexpression internally. If you're administering some sort of gene therapy or cell-based therapy, does the protein that you're intending to produce get produced, and where? And then, of course, you have the more broader category of pharmacodynamics. Does my drug have a biological effect at all? And then ultimately, the most important question, is there a response? Does it impact the course of disease in a meaningful way? And so for us as a community, the imaging tools that we utilize are really all aimed at just answering those questions. They enable us to measure, and in the case of imaging, to visualize biological processes and the drugs that we're using to try to manipulate that. Exactly as Antoinette pointed out, I'll make a couple of different points here. CFT sits within a broad range of technologies that we have available to us. There are advantages and disadvantages that this room knows as well as I do across all of these. In some cases, we're dealing with very high resolution technologies in vivo, like an MRI or a CT, that don't offer the sensitivity that we need to detect certain protein level or biochemistry level activity. Or we may have very high sensitivity PET and SPECT that do not have the resolution that we want to be able to look at small structures. Ultimately, I don't know if we want to agree it's gold standard or not, but at the tissue level is where we get sensitivity and visualization at cellular and subcellular levels that really tell us about proteins and receptors, that really tell us the biochemistry of what's happening in pathological tissues and in normal tissues. But as researchers, we do need tools that help us to bridge the two. And CFT offers an opportunity to interrogate a large amount of tissue at a high resolution and high sensitivity, despite being destructive. But as we've seen, as long as it's easy and it's quick, there's a lot of information that can be gleaned from this. I do not need to introduce what it is. I will say the user perspective on the workflow, the video that Antoinette showed is far better than the slide that I have here, but it is really that simple. It's about blocking an animal, putting that animal into the instrument, and then allowing the instrument to do its thing. So I want to just focus the rest of this on maybe a quick review of some applications. Rather than digging into one and really ripping it apart, I thought I would go from a less complex to maybe more complex example. Really beginning with, well, let's just see how something distributes in the body and how it clears. That's an important question for people to ask. As a CRO providing services, it's often about, well, how does the molecular weight of my compound influence its distribution? How does the charge influence its distribution? If we were to compare these 5 molecules, what do we see that's different amongst them? So biodistribution and clearance is not something that is ever completely solved because it will be molecule dependent. But if we start there and then move on to looking at target cells, talk about antisense oligonucleotide biodistribution, whole body, but then also locally, and then also get into some of the protein expression characteristics. So the last example is a viral vector, how it distributes in the body, and then the gene expression that subsequently occurs. So we're trying to really capture all of those important questions that are being asked in drug discovery. First example, and this is courtesy of Dr. Craig Ferris at Northeastern University, and he's trying to better understand for his applications the distribution of and clearance of molecules that are injected interventricular into the brain. And so what is done in this experiment is animals are Q dots are infused into the ventricles. The animals are sacrificed, blocked, and sectioned, as you have seen. What Antoinette basically just described is this is now a day's worth of work. So 5 animals can be injected. Those 5 animals can be blocked together. Those 5 animals can be sectioned overnight, and there you have 3D images the next day. I put the image on the right more for my own scientific purposes. When I looked at this, I was a little confused. It didn't occur to me at first, it should have, these are animals obviously, so olfaction is so important to them, but the nasal turbinates really stood out to me and it surprised me. So I did find an image from the literature from 2015. Badusi and all did actually kind of a similar experiment, but this is with labeled dextran. And you can see the nasal turbinates. The one point of reference here is that image is upside down, so the nose is down where it's up on the CFT images, just to help you out there. So moving on, an experiment that I'm sure many people in this room have done thousands of times, and that is to look at labeled cancer cells and then to treat those cancer cells. And often we will do this by bioluminescence or fluorescence. In this case, looking at ovarian cancer cells that have been engineered to both express TD tomato, but also to express the HER2 receptor. And so by fluorescence imaging, planar fluorescence imaging, we can certainly see that those cells are fluorescing. They're where they were injected. And on the right-hand side, a HER2-specific protein has been Alexaflor labeled and injected. And this fluorescence image shows really good correspondence between where the cells are and where the protein expression is. And in some cases, this is certainly good enough. To Antoinette's point, you've run this experiment, you're going to sacrifice these animals at the end, and certainly CFT in this case is going to provide a substantially more information than just the 2D image alone. I don't have the overlay of the two, but I will show some of the 2D data that Antoinette referenced earlier. It's really powerful. For those who know in vitro, we love 3D spinning MIPS, so you will certainly see plenty of those today. But there is a tremendous amount of information in those white light, I might argue maybe better called anatomical, reference images and the fluorescence overlaid with one another. But this is certainly going to be value add for anybody that's trying to compare the biodistribution of a protein to target cells. Continuing on the biodistribution front, and as we just saw, in vivo fluorescence has an important role to play here, and this would be a really common experiment for folks. So this is an antisense oligonucleotide that has been fluorescently labeled, and in an IVUS instrument, we're looking at imaging both prone and supine. So very common format to flip the animals over and look at both sections of the animal. At the end of this study, so you can see at the top, I don't know if you can read it, it goes from day 6 to day 15. That's what the D stands for there. At day 15, the animals are submitted for CFT imaging. So certainly from the in vivo, and one of the most important aspects of an in vivo study is that temporal information. So we're not just seeing it at a single time point, but each animal serves as its own control to see how it responds over time. It's super important here. The problem, and the obvious problem, with a 2D methodology is that any signal that sits below a brighter, more prominent signal is going to be lost. So in this case, some of what you might see from the lymphatic system will be obscured in a 2D view by brain structures or other structures. The other thing, if you can see in this image, is that you do not, because of the curvature of the spinal cord and the vertebrae here, there's actually a break in the image. And in this particular study, this ASO is infused intrathecally. So to understand the full distribution up that track is critical. And so losing a giant section of information is not good for the interpretation of this data. So although it does give you a time course in vivo, you're missing some pretty critical anatomy in doing that analysis. So the CFT is certainly really helpful here in understanding whole body. Staying with ASOs for for a moment, and now thinking about a more local administration. So Dr. Maraganori's comment about delivery. It may not be that you're going to administer intrathecally or systemically, but you're going to administer something locally. So in this case, interocular administration of an ASO. And not just, so in this case, that ASO labeled with sci-fi, but it's a transgenic animal for which where the ASO is present, you will get a gene expression. And so this gets to that point of, well, it's great to see where your drug product goes. Can we understand anything more about the PD associated with it? So on the left, you can see the whole body images associated with it. On the right, you can see the CFT at day 15. On the top right, relatively easy to see bright signal in one eye where this injection was made. Also, the body, there are, of course, naturally occurring fluorescent molecules in the body. So you can see porphyrin in the Herderian glands. So some knowledge of anatomy and biology is really important in what we do. On the bottom, it's a little bit more complicated to see what's happening from a protein expression perspective. So as I promised earlier, let's take a look at the 2D images. On the top, you're still seeing the labeled ASO, so the biodistribution of the drug itself. And on the bottom, you're seeing now that ASO-dependent GFP expression. So is it doing something while it's there? And in the 2D images, the white light, or as I was referencing earlier, these are really your anatomical references. So this is a built-in ability to see what's happening anatomically. You can see the injection site, and as I mentioned, Herderian glands also are lighting up here. On the bottom, you can see that there is protein expression only in the eye where the injection was made. So this compound is doing as it was intended. One of the examples I think that we've shown most frequently, so anyone who's seen us talk about CFT before will have seen this one before. And I did want to make another point here rather than just show it because it's a cool image, but the combination of the 2D and the 3D together. In this case, we're looking at viral AAV9 gene therapy and protein expression. So very simple experiment that was run very early on. If you have an AAV that drives GFP expression and you inject it systemically, what happens? Where does it go? What do you see? So certainly no one here who knows about AAVs would be surprised to see a tremendous amount of signal in the liver. This is exactly what we would expect to see. But if we interrogate the 2D images a little more, rather than just utilizing those 3D images, we know that AAVs each have their own tropisms. And in the case of AAV9, more than just nervous system and heart, but those are two of the tissues that they tend to home to and they tend to react in. You can see here evidence of DRG. And then on the right-hand side, certainly plenty of signal occurring in the heart. So when exploring AAV9 or any other AAV or modified versions of AAV, this can be a really fantastic technology to help first interrogate the whole body. Ultimately, we're going to go to pathology. We're going to do tissue-based sectioning. We're going to go at a higher resolution, obviously. But to interrogate entire panels of tissues and to gross out all those tissues and to spend all that time doing it, not sure where you're going to see anything, is frankly a bit of a waste of time. So this technology is bridging and enabling for additional interrogation. Last example, and then I'll wrap up, and I hope this wasn't too many examples at once, but I did want to kind of go from less complex to more complex. I showed you an image earlier of intrathecal administration followed by fluorescence whole body imaging and some of the limitations of depth for in vivo fluorescence. So certainly at in vitro, many other places, we use a lot of nuclear medicine imaging. So in this case, rather than fluorescent, we took the AAV, we labeled the capsid with I-125, administered it intrathecally, and then we could monitor the biodistribution up the spinal cord and into the brain. Because we're a CRO and because the brain was the most interesting anatomy to this pharma company, we are only allowed to show the bit that does not include the brain, unfortunately, where all the action is. However, I think the point to make here is that this is a combination of an in vivo scan at okay resolution, but very high sensitivity for biodistribution, and an ex vivo technique, CFT, where you're able to merge two 3D data sets. They're of wildly different sizes, and they're very different resolution. But it just becomes an image analysis problem at that point. So on the left, you can see the SPECT CT. We can see where the I-125 AAV capsid is residing. And here, right around the point of administration, where we can see where there is protein expression that's occurring by CFT. So we're able to visualize both the biodistribution and the protein expression in these animals. And on the right-hand side, you can see, as I mentioned, you can merge these. They're both in 3D space. So you're able to merge these with some image processing tricks. That may have been a lot of examples, but what I really wanted to do is to highlight that a technology exists and we have an opportunity to develop new methods, new applications on the back of it. And I tried to highlight that, really focusing on biodistribution and opportunities in protein expression or gene therapy, protein therapies. In the traditional space, so small molecule, large molecule drug discovery, that relationship between PK, PD, and efficacy is essential in advancing any molecule. It's a critical part of the drug discovery process. It's how you identify and molecules in the hit to lead process. It's how you identify your lead and then ultimately optimize it. And in that very important decision to move a molecule into the clinic, animal models really assist in that characterization and in that identification. It's a very expensive proposition to take a new drug to the clinic. So the more we can do preclinically to support that, the higher chance of success there will be. And certainly the distribution and excretion and clearance of a molecule is critical. I might argue that RNA, DNA, cell therapy is still relatively in its infancy. There are a lot of approvals. There's a tremendous amount of activity in this space. But biodistribution is critical. We all are, I'm assuming many of us are enjoying the benefits of RNA-based therapies right now. They just put it directly in our arms. But when you're going to treat more complex diseases, it becomes a much more difficult problem. And it's not just distribution and it's not just expression, but if you think about expression, it's both localization, where does it occur, but for how long and how strong. So there's a lot of unanswered questions that companies are going to have. We're here to help answer those questions, and we need technologies. CFT is one of those technologies. So I see it as a valuable tool, providing whole body, high resolution, high sensitivity information to us. It complements what we do in vivo and what we do in tissues already today. And as Antoinette pointed out, reduction in the use of animals and getting more information from the animals that we are using. And that Invigro is proud to play our role in the early conceiving of CFT as a technology, but Amit has really done a fabulous job of bringing that to life and doing the engineering and the science required to create a system that we can use now reliably to ask these questions, to see things that we were unable to see before, and ultimately to develop new applications. So with that, I will turn it over to Steve, and happy to take questions after. Thank you.